Okay, good afternoon, everyone, and thank you for the organizers for the opportunity to discuss this work, which was just recently published in Genome Biology. And so this is actually a slide that you probably saw yesterday in Julian's talk, and it kind of describes the traditional view of splicing, where, okay, we have these intronic and exonic splicing enhancers and suppressors that help regulate splicing, but from Hagen's work, we know that splicing is predominantly co-transcriptional, which provides an opportunity for chromatin structure to affect the process of splicing. And Mayan talked about this in her talk earlier today. So there's different ways in which chromatin state can affect splicing through the rate of elongation of the transcript by helping recruit other splicing regulators, etc. And well, how does this all happen? Well, what we're going to provide evidence for is that transcription factors and other DNA binding proteins contribute to this process. And just as a teaser, we looked at ChIP-seq data of some of the transcription factors that we are going to discover later. And what we found is that, well, they have a lot of peaks inside genes. In fact, the rate of peaks inside genes is, for most of these, is higher than intergenically. And here we just remove the first exon and intron and the promoters for this analysis. So maybe some motivation for actually thinking about this. And just a little bit of the history of how we started to work on this, and well, why intron retention? Well, I've been working with my colleague Reddy at CSU for over 10 years on intron retention in plants. And well, in plants, we didn't quite have all the data that we have for humans. And so, okay, we'll start playing with human data for intron retention. And by the way, intron retention in plants is the primary form of alternative splicing, and it's getting some more traction and love in humans as well these days. And around that time, well, a lot of you probably saw that Bassett paper that kind of revolutionized the way how we look at regulation of chromatin structure. And yeah, I happened to talk to David Kelly at an ISMB around that time. And I told him, well, what do you think about trying to analyze introgenic sequences with this? He said, I don't think this is going to work, but we still tried it. And this is what I'm going to discuss next. So we addressed a simple classification problem, trying to distinguish between retained introns and non-retained introns with a focus on those that overlap regions of accessible chromatin where the data came from this large collection that David Kelly assembled. And we used basically the same architecture that they used in Bassett. And well, we got pretty good results using this architecture that we just fine-tuned for this problem. And yeah, coming from a kernel methods background at the time, well, I compared to an SVM, and well, yeah, the deep learning worked quite a bit better. And what was Also quite interesting to us is that when we used a pre-trained Bassett model, it didn't improve performance. This is trained from scratch on this problem, which is from a deep learning perspective, maybe a pretty small data set. And by the way, the title of the slide, a little bit of a pun on, where did that, oh, there's a slide missing in this version of the talk. Yeah, I had too much time after the fun dinner yesterday, and convolution is all you need. So, well, what other, so there's this famous paper in deep learning called Attention is All You Need. And I look, well, do other people use this to name their papers? And there's pages upon pages in Google Scholar of various is all you need. Even Hopfield networks are all you need, and which is I don't believe at all. But anyhow, back to our topic. And so we next tried to look at, well, what is this network learning? So we did the usual thing, created motifs out of the filters, searched for matches for those filters. And what was really interesting is that most of the hits for those filters were transcription factors, not RNA binding proteins. So, and well, as you all know, this matching process is very error prone. So I'm actually going to show a somewhat even more convincing approach to do this. But this was pretty consistent across the various architectures that we tried. And we, of course, wanted to validate some of our predictions. So these are some of the transcription factors that our method picked, with a focus on the zinc finger ones. And so the the transcription factor occupancy measured from ChIP-seq data for these IR filters was much higher in the retained introns versus the non-retained introns, with the opposite for the non-IR filters. And well, our nice reviewers in genome biology also suggested, well, there's these knockdown experiments for some of these transcription factors, and we analyzed those data sets as well. And we looked at the introns that exhibited differential IR in those samples, K562 cell line. And so what we found is that, well, in the flanking exons of those retained introns, there's a big difference in the rate at which at the number of hits for those transcription factors, which is, again, further validations for the involvement of these transcription factors. Now, not a whole lot, we couldn't find a whole lot in the literature about these transcription factors and involvement in alternative splicing. The only one among these that we found information about was MAS. So, well, hopefully someone will do some experiments and maybe give some additional experimental validation about this. But then, one of the reasons it took us this long to get this paper published, we started this around 2018, was we took a little bit of a detour. We had a really nice idea how to use self-attention to predict cooperativity between transcription factors, and we applied it here and got plenty of predictions for that, some of which also panned out from ChIP-seq data. And finally, I'm going to describe some more recent results that are not in the paper. Well, kind of how would you do this using 2023 technology of deep learning? And well, nowadays we have all those fancy large-scale models of chromatin. Yeah, Bassett didn't really help us get better results, but so we looked at this model from the Zhu and Troyanskaya lab, where they have a model that from sequence gives you about over 20,000 outputs that describe various aspects of the chromatin state, including over 9,000 transcription factor and other DNA binding profiles. Well, some of these transcription factors are represented multiple times. Yeah, there aren't 9,000 transcription factors as far as I know in human. So the idea is to use the representation learned by this model in order to get a more accurate predictions of IR versus non-IR. And so that's what we did. And also what we did was to focus just on those. So what we did was to use the 9,000 or so predictions of transcription factor binding events, use that as input to very simple logistic regression. And that avoids the big problem we had in our previous approach of having to map things to transcription factors. And so here we can directly implicate the relevant transcription factors in the process. And the other nice thing about it is that this whole model, rather than hours, took just 30 minutes to train. And here we got lots of, so it's very easy to analyze the weights of logistic regression and get the relative weights of different transcription factors. And in our top 10, we got some of these proteins that, yeah, all of them are documented in the literature as having some involvement in alternative splicing. And yeah, and probably those of you who know a little bit of the biology know that this is not a HDAC6 is not a transcription factor. It's a histone deacetylase. So, but that's also relevant to this problem. So, in conclusion, we got better accuracy using this logistic regression approach than our initial model. And actually, if we use the raw output of, say, rather than the internal representation that it uses rather than the output, then we're able to get even better performance, but we lose a little bit of the interpretability. So in conclusion, what I hope to have gotten across is that transcription factors and other DNA binding proteins are major players in regulating alternative splicing. So we need a better understanding of how this works. And in future work, we're planning on, this was very simple, retained, non-retained. We need to do more work on doing this in a condition-specific way and maybe try other forms of alternative splicing. And then, hopefully at some point this work will get funded so we can actually work with Mayan to get some experimental validation. And with that, I'll thank my collaborators for this work. So this was Fahad's PhD thesis. And this is in collaboration with Mayan and my colleague Reddy at Colorado State. And with that, I'll take your questions.
Hi, is there anything unique about these introns that are retained? Size, splice sites?
Well, what we know about retained introns is that, yeah, they tend to be shorter. They typically have weaker splice sites. So there's a paper from a few years back that it's called something like the intron retention code. And yeah, they discuss in detail the differences between retained and non-retained introns.
And are they more situated in the 5' end of the gene, or do they potentially have alternative transcription start sites?
Yeah, I don't have a good answer for that.
So I have one more question. Well, what do you think might the model be? So, you know, you have a transcription factor binding in an intron. How does it actually then lead to intron retention?
Well, what it can do is attract some splicing regulator. It can affect chromatin structure directly, and that indirectly affects splicing. So yeah, there's different ways in which this could work, and yeah, we're hoping for yes.
It seems as though that that there was some specific transcription factors that showed up, and not generally all transcriptions. So
Correct. Yes.
Would point to some, let's say, protein or structure-specific regulation that does something very specific, rather than
yes.
just opening, closing chromatin or something.
Yes.
Some retained introns are hosts for SNRNAs? Are those
I don't know.
Okay. It could be interesting to look at that.
Yes.